2, 3. Hi and welcome to Attempting Science, LIU iGEM podcast. We are at Mjärvik Creative podcast room, and I'm Emmanuel. With me today is
Alexandra.
Yes, hi and welcome.
Thank you.
Yeah, you're also a team member of LIU iGEM. And maybe tell us a bit about yourself. What do you study?
Well, I have just finished my second year at the biomedical engineering program at Linköping University.
Really cool. Yeah, and you're also a team member of LIU iGEM's Human Practice Group.
Exactly.
Yes, so maybe we should start. Our theme today is gene editing. And also we're going to talk about a bit of cool examples, some interesting tools, and some ethical or some dilemmas with this tool. Yes. Would you like to start? Maybe we can give some examples of these tools.
Yeah, we are mainly going to talk about 3 different tools, namely CRISPR-Cas9, zinc finger nuclease, and TALEN.
Yes.
And I thought I should start with CRISPR-Cas9.
Yeah.
And well, CRISPR-Cas9 is the most, it's one of the most usable, well-known, well-used, I should say, one of the most well-used methods today, because it's cheap, it's fast, and it's more accurate than a lot of the alternatives.
Yeah, exactly. It's much more easier to design because of its what do we call it? It's building blocks, building blocks, so to speak. Yeah, and maybe we should talk about those.
Yeah.
Because it has Cas9 and guide RNA. Maybe you should explain those. I put you on the spot, you know.
Yeah. Feel violated. Well, the Cas9 is an enzyme, and as we should probably mention, an enzyme is protein that can start some sort of reaction or cascade in the body, or in a cell, or so on. And the RNA is a guide RNA. It's called guide RNA or gRNA. This helps the Cas9 to find the right place in the DNA to cut the sequence.
Yeah.
So the guide RNA can find specific spots or sequences in the DNA strings, and the Cas9 helps cut it.
Yeah, exactly. It has a correspondent part to the gene that it wants to edit. So if there's an A, there's a T on the guide RNA, and so on. Just to have a perfect match. So romantic.
So romantic.
And yeah, the CRISPR-Cas9 also needs a PAM sequence, which is a sequence just to make sure it's on the right place. Without it, it can't bind in, so to speak. Yeah, and it's the tool with the brightest future at the moment. But yeah, there's a lot of research on other, because CRISPR-Cas9 as anything else has some some negative parts on it, of it. But we will go into that later on. And yeah, 2 more tools. These are TALEN and zinc finger nucleases, or shortened by ZFN. And they're a bit similar, right? They have this FOC1 sequence. I'm not swearing, it's named that. And this FOC1 is a protein that's also similar to the Cas9, which cuts the DNA. But the difference between these are the proteins that recognize the site where it should be. Do you want to talk about those?
I'm not sure if I have it written down.
Yeah, I can. It's the zinc finger.
Yeah, the zinc finger protein.
Yeah,
Yeah, well,
on the zinc finger-like nucleases. a bit,
Oh.
a bit obvious, maybe.
perhaps. Well, zinc finger is a protein.
Yeah.
It's characterized by one or more zinc ions.
Yeah, exactly. And the thing with zinc finger-like nucleases are that they aren't like the guide RNA where they detect one single base pair of the DNA at a time. It recognizes 3 at a time. So it's 3 base pairs are called codons. So they detect multiple codons. And the taulen has, instead of these zinc finger proteins, they have tau effectors, which are also proteins, but they detect each base pair with these proteins.
Yeah, and these they can quite easily be engineered, right?
Yeah, quite not as easy as Cas9, but a bit easier than zinc fingers proteins. Yeah. And these are the main. There's also others like CPF1, I think it's called, and also let's see, Lambda Red, for example, and others. But these are the most well known. So, yeah. And also, the funny thing is that our iGEM team won't use any of these. Because we, as we said before, are going to gene edit bacteria to create a new strain with by inserting genes. But we're going to do something called double crossover, which is instead of cutting, we just put in a sequence of DNA called a plasmid. That's a circle of DNA, and it will have matching parts to the bacteria genomes. So it will be integrated by the bacteria itself. So we will fool it, so to speak. Yeah.
So we think outside the box here.
Yeah.
In Linköping.
Not we, but a team member read upon it, so we wouldn't take any glory. We might have to change it, but at the moment it seems like the best alternative because, as we said, CRISPR-Cas9 has a bit of downsides. For example, it might cause mutations, and it can't have. If we are going to have an insertion of a gene, we can't have super much DNA, so to speak. Yeah. So that's our motivation.
Yeah.
We might be wrong, but that's how we wanted to do it. So.
Yeah, there is a lot to consider, and we have tried to do our best to come to a reasonable conclusion.
Yeah, should we go to different cool things you can do with it?
Yeah.
With gene editing.
Yeah, well, gene editing is very applicable.
And sorry, I forgot. We can say that there's different kinds of gene editing also. You can either remove a gene completely. You can either, which is called a knockout. You can also do a knock-in, which is that you put in a gene and that express, for example, a protein or something. But you can also put in a gene that might silence another gene. So there's so many options of how to do it. And also you can just put in a slight mutation, maybe change the base pair or something and change the whole sequence. So there's a lot of considerations.
We can do a lot.
Yeah.
With gene editing in many different ways.
Yeah. So yeah, as we said before, cool examples.
Yeah, and as we said, like it's very applicable in general. We can use it for just editing genes or genome or embryos.
Yeah.
But it can also be used for example, to treat diseases.
Yeah.
And research and so on.
Yeah.
We have
and for example, also GMO, gene edited food instead and stuff like that.
Yeah, exactly.
So one example is that you can gene edit immune cells. And immune cells are cells that protect the body. They attack pathogens and stuff. So what you can do if you have cancer is that you can gene edit a certain immune cell called T cells. So you edit the cell to be extra sensitive to cancer cells. For example, the cancer cell has different proteins on the outside, different like flags, and receptors with which is like a mailbox. They receive messages and activate certain things. So, for example, cancer cell has a lot of growth receptors that they are extra sensitive to some proteins to make them grow. So you can gene edit these T cells to be extra sensitive for different kind of cancer-coupled proteins, and then you can put these edited T cells back, and then they will be extra sensitive to the cancer and be more applicable to kill them. So T cells are super good at killing stuff, so it's a really good tool to use. So they will really target the cancer, and those people who it has been tested on has had I think lymphoma and bone marrow cancer, and those persons has had months and years of what's it called, regression. Yeah, I think so, of the cancer. So it seems to be a really great tool. And for these persons, the chemotherapy didn't work. So it's really a good example of maybe something to use as a last alternative or together with chemotherapy.
Yeah, so we probably use the body's own adaptive immune system
Yeah.
in order to kill a very, very harsh disease.
Yeah.
And hopefully it will be developed, and yeah, hopefully we will be able to use it even more in the future.
Yeah. And do you have something cool you found out? We had a bit of homework last night to find different cool stuff.
Yeah, I found an interesting study about eatable vaccines.
Cool.
And these vaccines are engineered. They are, well, it has been a few articles about, for example, to replace shots or treatment with bananas was a
Super bananas.
super bananas. And that was an article about how we can use gene-modified bananas with vaccine for hepatitis B in them, in order to, well, in, for example, countries where the healthcare system is quite reduced. So instead of going to the hospital and get a vaccine shot, you can eat a banana and get practically the same amount of vaccine in your body.
Cool.
And it's a great thing in theory, I think. It's a great thought.
Yeah.
But it has had some, well, there are some concerns regarding the practical use of it,
Yeah.
of this method.
Yes, because as we have talked about, the vaccine consists of different proteins called antigens, and these antigens are like different parts of a pathogen, for example, a bacteria or a virus. So instead of introducing the virus, you introduce its parts. So the immune system will react on these parts when the real bacteria or virus enters the body. So maybe it won't survive the stomach, or what's the problems?
Yeah, well, it's a question of if it degrades at the right place in the body,
Yeah.
so we can get the use of the vaccine, or if it just is disposed again and not helping us at all.
Yeah.
I think. So yeah, well, hopefully this will work someday.
Yeah.
But it's a quite old study as well, so I'm not sure where they are with this technique today.
Mm-hmm.
But 10 years ago, they were positive.
Yeah. Maybe they're still studying. Who knows about?
Yeah,
it.
hopefully
Yeah.
they are.
Yeah.
Because it's, as I said, it's a great.
Yeah, because otherwise you need to have the vaccine bottles and medical staff and
Sterile
yeah,
environment and so on, which could be quite hard to get in some countries.
yeah, exactly, and also just transports the stuff and also how long shelf life they have. So there's a lot of factors that might that the edible vaccine might have a better chance of. So yeah.
Yeah, hopefully.
Yeah.
And maybe we can, I don't know, maybe we can treat the bananas so they, so that they like have this vaccine while growing as well.
Yeah.
Perhaps. We'll see.
Yeah. And yeah, different things that has been treated with also gene editing is other genetic diseases. For example, sickle cell disease, which is a disease where the red blood cells, the cells that delivers oxygen to the tissues, are defective. They have the wrong size and shape. They're also called erythrocytes. That's difficult to say in English. So what they have done is the red blood cells are produced in the bone marrow. So they've taken out the sick bone marrow, not everything, but a part of it, and gene edited those cells that later on becomes the blood cells, so that they don't have this defective gene, and then they eliminate the bone marrow in the sick person, the patient, so they don't have anything, and then they reintroduce the bone marrow that is now edited and should be correct. So then it will grow and probably produce correct blood cells. And after a 2-year checkup of one patient, it seems that he has enough healthy blood cells to not have any symptoms of his disease. So it's also a really cool way to use gene editing.
Yeah, it's a great way
Yeah.
to use it, I think. I mean, isn't that like the purpose of gene editing, to be able to do such things as this example?
Exactly. One thing is maybe edited earlier, for example, in the embryo, but not everything is detected in an embryo. So this is a really good method to do afterwards, because in theory he shouldn't have any sick blood cells left, only healthy ones. So it's quite risky to just empty a person of the bone marrow. But if it succeeds, it's great. And as I said before, after a 2-year checkup, everything seemed okay.
Yeah, and while doing this procedure, they are quite vulnerable and sensitive to the environment as well.
Yeah, exactly, because it isn't only blood cells that are produced in bone marrow. There are also immune cells that are produced there. So you're really sensitive to diseases. So you need to be in a sterile environment during that time. So quite lonely.
Yeah, quite lonely, and quite hard to keep a totally sterile and environment around the person as well.
Yeah, and some people die. It isn't 100% safe. So it's quite a risky procedure.
And how's the success rate at this?
I've only read one published article on this, and then it was a success rate, but I don't know otherwise. And it also depends on the country and the staff, how well educated they are. So that's why it might be more difficult in countries that has a bit more problems with their healthcare. So yeah.
So is it like a common procedure to do, or?
No, not yet at least. Bone marrow, just replacing bone marrow and not gene editing is not super common, but it happens. but not when you remove the bone marrow and gene edit. It's not common, because now you can either completely eliminate the bone marrow and put back your bone marrow, or get it from a person that has healthy bone marrow and just transplant it. But those are also really risky procedures.
Yeah, bone marrow in general.
Yeah, it's not something to play around with.
No, not really.
No.
But it's a great thing that it can be done
Yeah,
if there is no other way of being treated, so to speak.
exactly. There has also been tests on embryos, and they have, for example, with beta thalassemia, that is also a blood-related disease, and they have been able to delete that or edit that mutation. But it's much more riskier in embryos, because this procedure was with CRISPR-Cas9, and CRISPR-Cas9, as other gene editing tools, can cause mutations, because it can hurt other genes or cut it wrong. So embryos are super sensitive to that, and it might even worsen the disease, or maybe create a new one.
Yeah, it's not only the procedure that is risky with embryos. It's also quite ethically questionable.
Yeah,
So
because an embryo can't consent. uh, but you can also see it as, it's either that, editing, or having a super horrible disease,
yeah,
or maybe a deadly disease. But the question is, how deadly or how damaging should the disease be to gene edit? Should a gene that increase the risk of a certain disease be edited, or should a disease that will or a gene that will cause a disease edit, and stuff like that? It's quite. I don't have any answers, but there's a
No.
lot of things to consider.
Yeah, it's really hard to draw a line where considering what's okay and what's not, and probably each individual draws the line differently.
Yeah.
So it's nothing it's not right or wrong. Anything isn't right or wrong, but it's hard to yeah.
Because also
Being-
different countries has different systems. In
Yeah.
Sweden, healthcare is basically free, so in theory, everyone could have a gene edit if they would need it. But in some countries, maybe you need to pay for it, and then it can be a question, of social economic status and stuff like that, and that might not be fair. To just some people can have healthy children and some may not, and stuff like that. So it's a lot of things that need to be answered. I
Yeah,
think.
and that's already quite a big problem
Yeah,
already today,
exactly.
but for different reasons
Yeah.
in different countries.
Yes.
So it's a hard question,
Yeah,
and there is no right or wrong answer really.
it's really a personal answer, or how to call it. It depends on what might be religion. It might be just life. Your own opinions, maybe political opinions, and there are so many factors that might play a role in this. And also, should it just be for disease, or should it be for other stuff? Some people might want to do it for other things, and is that then okay or not?
Yeah, it's hard to make all of these different reasons. It's hard to make them cooperate
Yeah.
with one another. It doesn't work.
Yeah. Yeah, and also we discussed it before, me and Alexandra. There has been 2 published articles this year, I think, that seems to prove that CRISPR-Cas9 might increase the risk of cancer in gene editing, and that's because when CRISPR-Cas9 cuts both the DNA strands and that activates the cell system to say, yeah, this isn't right, there's a damage here. And it activates a protein called p53, and it will either shows if the damage is too great to go into cell death, apoptosis, or into a pause and repair mode. And none of these alternative is good when you need to use CRISPR-Cas9. But some cells might have damage to p53, which might cause the cell to neither go into apoptosis, cell death, or into a pause and repair mode. And this is much more better for the CRISPR-Cas9 system, because then it can gene edit. But the problem is that almost all cancer cells, like 50% of those, depending on the type of cancer, has this kind of mutation in the p53. And so it seems like the best cells to edit are cancer cells. And in that case, almost, according to these articles, those edited cells are the most cancer-prone cells. It's not 100% cancer cells, but it's a much higher chance to be a cancer or cancerogenic cell. So instead of maybe transplanting a healthy cell into a person, you transplant a cell with quite a high risk of cancer that is gene edited.
Yeah, and cancer is something we want to avoid.
Yeah, exactly. And
most days.
Yeah, and CRISPR Therapeutics and other CRISPR stocks dropped dramatically after these articles. There was 2 articles by Karolinska Institute and Novartis. But yeah, we need to remember these are only 2 articles and quite young or newly released articles. So they might just have found a super extraordinary example, or they might have some incorrect information, or they might be right. So it really needs to be studied more, because if that's the case, maybe we should go with a different method, because it seems to be just the CRISPR-9 system and not Tallen or zinc finger or something like that.
Yeah, so it's important to remember that these studies are quite new, and hopefully what they suggest can be, proven wrong.
Yeah,
so to speak.
hopefully. Not that we CRISPR or Karolinska Institute is in Sweden, and so we want to be a bit patriotic, but maybe not for this reason, because it's if that's the case, it might be quite a dark future for gene editing.
Yeah. Well, it's at least it's good that they have found it.
Yeah, exactly.
So we don't wander in the dark anymore if that's the case.
Yeah.
Because then we can consider it and.
Yeah. Maybe we can find a new, different kind of technique, or maybe you can edit the CRISPR-Cas9 system in some way to avoid this, because it seems like the double-strand break is the difficult part. So maybe if it can edit that, but yeah, we'll see.
Yeah, hopefully it will turn out all right in the end.
So maybe we should start to end this podcast. We might have woken some questions, but maybe not many answers, but maybe that's for listeners to find. So we want to thank you for listening, and this was the Attempting Science podcast with Leo iGEM. I'm Emmanuel, and with me today was
Alexandra.
So thank you, and feel free to come back and listen again. Bye.
Bye.